Hello, in this video we will look at the competitive ELISA test. And their uses, we will also discuss about that. So that means the competitive ELISA means the competition between the basically enzyme linked antibodies and antibodies which that is come from the patient. So the concentration of antigen we can detect, or concentration of antibodies we can detect which that is present in the patient. For example, here is the patient which we will take the sample from suspected person. So this suspected person contain antibodies which that is against the antigen. So it means we required a antigen in this case. So here is a serum which that is contain a sample containing antibodies. So we should need a antigen in this case. Why? Because we are taking the antigen from the lab to check that either present these antibodies against this antigen or not. So for example, the HIV antigen are present in the human being. So we can detect by our antibodies which that is produced against that antigen, for example HIV. So for example, we determining the antigen of HIV antibodies. So that antibody is basically antibodies come from the patient or suspected person. So here is the antigen in laboratory which that is developed. So this antigen. So this is the antigen is basically HIV virus protein or something you can say. So here is also one more important thing is the enzyme linked antibody which that is present, and it is also produced from the basically from the lab, and attach the enzyme is the conjugated antibody enzyme linked. So this enzyme linked antibody is here which that is look like that, and a antigen which that is attached from the attachment region, and Fc portion contain of this antibody is an enzyme linked. So enzyme can be horseradish peroxidase or beta galactosidase or alkaline phosphatase enzyme which that is used for it. So let's begin to understand about the enzyme activity. So here is the enzyme horseradish peroxidase enzyme, for example, which that we will add the substrate. So substrate can be TMB, tetramethylbenzene. This tetramethylbenzene, for example, will attach with the enzyme from the active side to bind the substrate to convert that product, and this product is show blue color in the spectrophotometer. So I hope you make sense about this mechanism. So let's begin to understand the sample serum containing antibody as well as enzyme linked antibody are present in this microtiter well. So this microtiter well contain a antigen. This antigen is come from the lab, and a antigen, for example, and coating buffer which that is protect the of the antigen to releasing. So that is important, and we will add the sample serum containing antibody which come from the patient which that is a suspected, while on the other hand enzyme linked antibody is add. So we can compete both antibodies against the antigen. So here I am drawing the two microtiter well to understand. So first of all, we will put the chromogenic substrate for showing, for example, the tetramethylbenzene. So here is the antibodies is attached more with the antigen which that is come from the suspected person, while the most of the antibodies bind from sample and enzyme linked antibody is attached less. So it means the color will show after the product is less. So it means we can say that the person is suspected and person is confirmed of the HIV infection due to the producing of antibodies against that HIV virus. So, on the other hand, the most of the enzyme linked antibodies attach with the substrate. For example, so it means the less amount of antibodies is attached with the antigen, so it means the more color will be produced. So, if color will be produced more, it means the less suspicion or it is the less amount of the antibody are present. So, we are checking the person containing more or less antibodies with respect to the immune system. Either immune system is good or not. So here we will add first washing. After the washing, the extra antibodies will sloughing off or shedding, while the incubation in certain time during that certain time and after this washing mechanism to release that antibodies which that is wasteful for us. For example, that is not attached with the antigen because antigen is completely binding with the antibodies which that is enzyme linked or antibodies come from the serum or sample. So it means the important thing is that the more absorbance value or less absorbance value can be occur. Why? Because addition of chromogenic substrate incubation, addition of strong acid to stop it. So after the stop, the strong acid will be added, so the reaction stop. So after the reaction stop, we will check the spectrophotometer to check that color. So here is the more absorbance. Why? Because the more enzyme linked antibodies attached with the antigen. So that is why it is the color will be more. It means the antibodies are present in the person is less. So it means their immune system is not. Or on the other hand, if not attached, the more color will be shown. It means that person is not suspected with the HIV virus. So we will use another antigen to detect what is that. So the less absorbance mean the more antibodies, which it means the confirmed the suspected person. So the less absorbance is the concentration of antibody in same sample is more. So the more absorbance is the concentration of antibody in sample is less. So here is the graph. We can understand well with the graph to understand the absorbance value. So the concentration of antibodies on the other hand. So the horizontal portion shows that the milligram per ml concentration of antibodies, while on the other hand the vertical is the absorbance value. So the 0.00 to 1.20 is the absorbance value which that is show the color. So for example, when the concentration of antibodies is more in the serum, it means the color will be produced less. So you can see in this diagram to understand as well. So that is important thing. So if we check the antigen in the infected person, we uses antibodies instead of antigen from lab. So we can use the antibody for checking the antigen, that is the vice versa, and it is the opposite system. So the mechanism is say, so that is the competitive ELISA test which that is compete against the antigen with the enzyme linked antibody as well as serum containing antibody. So thanks for